going to start. So welcome everyone. We are on another Solanaceae seminar online this Friday. And I'm very glad to introduce Marcela Moré today from Argentina. So Marcela completed her doctorate at the Universidad Nacional de Córdoba under the supervision of Andrea Cocucci and Alicia Sarsic. Her doctoral dissertation was focused on pollination by nocturnal hawkmoths, Lepidoptera sphinxidae, in native plants with extreme flower lengths from subtropical Argentina. She performed a postdoc under the supervision of Robert Raguso, studying the evolution of flower traits mediated by pollinators in the genus Jaborosa. She undertook internships with Rodrigo Medel at the Universidad de Chile, with Robert Raguso at the University of South Carolina, and at Cornell University, and also with Ian Kitching at the Natural History Museum in London. Currently, she is a CONICET researcher at the Instituto Multidisciplinario de Biología Vegetal, or Multidisciplinary Institute of Plant Biology in Argentina, in Córdoba, where she studies different aspects of the pollination ecology of plant species native to Southern South America. So with that, I just going to pass you Marcela. Thank you very much for today. And she's going to talk about the Corolla color and floral scent evolution in Jaborosa, of course.
Thank you for the invitation, Rocio. I'm very happy to be here and share you my work on Jaborosa. I will share the presentation. It's okay?
It's still all black, but I think it's coming. Let's see if you, because we can see the pointer, so if you click, it show up.
It's at the end.
That's why. Yeah.
I will. Almost there. Okay. It's okay there? Yeah.
It's not sharing now.
No?
No, you have to share screen again, I think.
Okay.
Mm-hmm. Now it's going. Yep, perfect. Perfect.
There? Yes. Okay. As a pollination biologist, I was thrilled by the amazing diversity in flower morphology, corolla color, and floral scent present in Jaborosa. I started studying the genus during my postdoc and the supervision of Robert Raguso in collaboration with Andrea Cocucci, Alicia Sarsic, and Gloria Barbosa. Today, I am going to show you some results in relation to the evolution of two floral traits that are important in pollinator attraction: corolla color and floral scent. As a brief introduction, the rise of the Andes is considered to be one of the most important events that promoted the diversification of plants that we observe today in South America. And this is because the Andes act as a north-south dispersal corridor, as a barrier, preventing the dispersal of organisms both between the east and west regions, as well as between isolated internal valleys, as a source of new high mountain habitats, and as a promoter of diversification in other regions bordering the Andes. There are several examples of Andean clades in which diversification occurs in concert with adaptation to changing pollinator environments. Here I show you as example the evolution of flower traits in the nightshade genus Salpicroa that was studied by Ana Clara Ibáñez, a former doctoral student in our lab. She found that flower diversification occurs in concert with shift in pollination modes. In this genus, the elongation or shortening of the corolla tube and the emission of floral fragrances are associated with the pollination by two main groups of animals, moths and hummingbirds. The genus Jaborosa comprises 22 species that exhibit remarkable interspecific variation in floral traits. Phylogenetic reconstruction based on 4 plastid and 1 nuclear region strongly supports the monophyly of the genus, with species resolved in 2 major clades. A lowland clade comprised of 3 species with nocturnal flowers that have white corollas and elongated tubes that produce exquisite perfumes and abundant nectar, and an Andean clade composed of the remaining species, which strongly differ in floral phenotype. Several of these Andean species have diurnal and rotate flowers with maroon to black corollas that emit a stinky smell and do not produce nectar. The split between the lowland and Andean clades possibly occurred 10 million years ago in the late Miocene, with recent species diversification in the Andean clade co-occurring with the last uplift of the Andes 1 to 3 million years ago during the Pleistocene. The genus is distributed from southern Peru and Bolivia to Tierra del Fuego in Argentina and Chile. The lowland clade, represented here with black circles, is distributed in lowland grassland below 1,000 meters of altitude in Paraná and Uruguay river basins and in the western Chaco region. The rest of the species of the Andean clade, represented here with white circles, mostly grow in the semi-arid regions located either at high altitudes, about 3,000 meters in the Andes, or at high latitudes in the Monte Desert and the temperate Patagonian Steppe. We recorded 2 very different pollination modes associated with these extreme phenotypes. On the one hand, there are species with rewarding flowers that produce abundant nectar with long corolla tubes, white corollas, and nocturnal emission of floral scents that are pollinated by nocturnal hawkmoths. On the other hand, there are species with rotate flowers that do not produce nectar, have dark corollas, and diurnal emission of fetid odors that are pollinated by flies. These flies are deceived by the flowers that use visual and olfactory cues to imitate the oviposition substrates. Between these 2 extreme phenotypes, there are species such as Jaborosa reflexa with an intermediate floral phenotype that have generalist pollination mode by small insects, including flies and bees. Nocturnal hawkmoths require a combination of visual and olfactory cues to find flowers in the wild. Although they rely on corolla color and floral scent to find the flowers, other cues may help in the task, such as the presence of carbon dioxide and humidity to detect rewarding flowers. Mechanoreceptors on the proboscis help them to find the entrance to the corolla tube. Gustatory receptors of the proboscis evaluate nectar quality, and antennal mechanoreception and vision control their hovering flight position during feeding. In the case of flies, it's mainly the presence of fetid scent that cue the trigger flower visitation in Jaborosa. Later, I will show you some bioassays we undertook to evaluate the role of corolla color and floral fragrancy in focal species. We recorded saprophyllous fly pollination in 5 Jaborosa species. Flies mostly belong to the families Calliphoridae, Musidae, and Sarcophagidae. Okay, so far it was an introduction to the study system. Now I am going to show you some results in relation to the evolution of flower color, taking into account both the geographic and ecological context. This study was done in collaboration with 2 doctoral students working in the lab, Ana Ibáñez and Eugenia Drevniak, and together with Andrea Cocucci and Robert Raguso, and it was published in a recent special issue of Flower Color Evolution in Frontiers. The expression pollinator climate was coined in 1965 by Grant and Grant, who stated that latitudinal or altitudinal differences in pollinator abundance are to floral phenotype as environmental factors are to vegetative phenotypes. Given the floral diversity and the geographic distribution of the genus, we expect that flower evolution reflects ancestral movement across contrasting pollinator climates. In this study, we accounted for the geographical context by collecting occurrence data from those species that we were able to measure corolla color. Occurrence data were classified in 3 environmental zones according to the ecoregion classification proposed by Olson, and are shown in the map in different colors. Lowlands in herb green, subalpine in olive green, and alpine in brown. Pollinators mediate a multidimensionally biotic selective context through their own integrated phenotypes, including different cognitive and behavioral abilities to find and visit the flowers. The 3 pollination modes are indicated with different colors: nocturnal hawkmoths in pink, saprophyllus flies in black, and generalist by small insects in green. In gray, there are those species where we couldn't record pollinators. As you can see, there is a clear transition in pollination modes from hawkmoth pollination in the east to saprophyllus fly pollination in the west and the south. The same colors will be used in the next graph. We measure the corolla reflectance in these 11 species, encompassing these 3 pollination modes. The x-axis represents the wavelength, range from 300 to 700 nanometers, and the y-axis represents the corolla reflectance measured with an spectrophotometer. Species from the lowland clade, Jaborosa o'donnelliana, roncinata, and integrifolia, show corollas with maximum reflectance between 450 and 700 nanometers and absorbing in the UV region of the spectrum. These flowers are perceived as white by humans. Diurnal species from the Andean clade show corollas with very low reflectance. 4 saprophyllus fly-pollinated species were maroon to black in color as perceived by human vision: Jaborosa laciniata, leucotrica, rotacea, and magellanica, or pale freckled with dark blotches in Jaborosa sativa. 2 species of the Andean clade show interspecific variation in corolla color. Jaborosa laciniata was polymorphic, with flower color varying among individuals between black or more rarely green. Jaborosa reflexa was ectomorphic in flower coloration, with individuals showing flowers of the same age, either yellowish to pale green or maroon. We examine overall differences in reflectance spectra among Jaborosa species using principal component analysis, which allow to summarize information of complex spectra in a few variables. The analysis was performed with standardized spectral data that removes total reflectance as a dominant variable and allows comparing only spectral shape. In the upper graph, you can see the variation in spectral shape among all individual measures measured. The first 2 axes of the analysis accounted for more than 95% of total variation. To explore the evolution of corolla spectral shape in the genus, we represented overall variation in corolla color among species in a phylomorph space here. You can see that variation in reflectance spectra is associated with clades, with PC1 loading scores clearly separating the 3 species from the lowland clade, from the rest of the species in the Andean clade. We also estimated the ancestral reconstruction for corolla color and pollination mode. Color is represented in the y-axis by the PC1 score from the previous analysis. Diversification within the Andean clade is associated with the pollinator shift from nocturnal hawkmoth to saprophyllous flies and occur in concert with changes in corolla coloration from white flowers to maroon and black flowers during the initial diversification of Jaborosa ancestors across southern South America. So far, these results show an objective variation in corolla color measured as spectral shapes, making no assumption about the receiver's visual system. To study flower color in pollinator subjective way, we represented flowers as loci in perceptual spaces using visual modeling. The light reflected from the flower is assumed to excite each receptor type according to this formula that calculates the receptor responses or quantum catch scores, where I is the spectral quality of incident light, which is daylight or twilight according to different pollinators. S is the reflectance spectra of several individuals of 3 glandularia species that looks white, purple, and red to the human eye, just an example. And R is the sensitivity of the pollinator eye. Many insect pollinators, such as bees and moths, have a trichromatic vision system with 3 light-sensitive opsins in the UV, blue, and green wavelengths. Our own vision is also trichromatic by sensitive to blue, green, and red, RGB. Thus, we calculated the quantum catches of Jaborosa species for main pollinators, nocturnal hawkmoths and flies. Color loci of Jaborosa flowers were represented in the visual space of the nocturnal hawkmoth, Manduca sexta. Each corner of the triangle here corresponds to one of the 3 photoreceptor types of the moth eye, UV, blue, and green, and the location of a color locus in the triangle represents the relative excitation of the 3 receptor types by the floral sample. We use the green leaves as background that appears in the middle of the central space. As you can see, the 3 hawkmoth-pollinated species were clustered together in the region between the blue and the green photoreceptors and distant from the central zone. Thus, they would be perceived as colorful objects against the leaf background by hawkmoth. We also calculated chromatic contrast between pollination modes that are shown here. F is fly-pollinated, H hawkmoth-pollinated, and X generalist. The highest chromatic differences was observed between hawkmoth and saprophyllous fly-pollinated species, and between hawkmoth-pollinated species and the generalist Jaborosa reflexa. No differences in chromatic contrast were detected among species pollinated by saprophyllous flies and the generalist species. Here. We also represented the color loci in the categorical color vision system proposed by Troche, based on the relative excitation of the 2 pale type and 2 yellow type receptors of a fly. The receptor of each pair that is stimulated more strongly determines which color the fly perceives. Thus, flies perceive 4 color categories that are represented in the 4 quadrants here: fly UV, fly yellow, fly purple, and fly blue. Floral color loci for the saprophyll fly-pollinated Jaborosa species all fall within the fly-UV quadrant, and carrion flies wouldn't be able to distinguish among them. Hawkmoth-pollinated species fall within the fly yellow and are distinguished from the saprophyllus fly-pollinated species. Finally, we use a phylogenetic generalized least square correlation to test whether phenotypic transitions across pollination climates occur along perceptual dimension of the changing pollinator environments. The x-axis represents the biome liabilities of the occurrent data according to the ecoregion classification I previously mentioned: humid lowland, dry lowland and foothills, and alpine vegetation. The y-axis is the color as perceived by hawkmoths and was constructed using a principal component analysis of the quantum catches we calculated using the visual modeling. Thus, the score from the PC1 were used as color vector values. We can conclude that evolutionary transitions along the color vision axis were significantly associated with the ordered transitions in environmental occupancy across the phylogeny of the studied Jaborosa species. In this part of the talk, I will show you some results in relation to the evolution of flower scent, chemical communication via floral scent plays a central role in attracting pollinators and is considered a key innovation promoting pollinator shift and diversification in flowering plants. When we first started studying Jaborosa fragrances with Robert Rauso, he identified the volatile organic compounds present in these two species. We were surprised because they were chemically simple. Jaborosa rosacea, stinky scent, was dominated by two oligosulfide compounds, dimethyl disulfide and dimethyl trisulfide, characteristic of carrion or carnivore feces. These compounds are commonly found in other species of plants with deceptive flowers to attract flies that use carrion or carnivore feces as brood sites. Jaborosa integrifolia has a delicious scent that is dominated by four compounds: benzaldehyde, benzyl alcohol, methyl salicylate, and farnesol. We first did some simple bioassays to confirm whether these fragrances play a role in pollinator attraction. We use artificial paper flowers and a scented bait with these compounds, and we were able to attract calyptrate flies to the black paper flowers baited with oligosulfides here, and Manduca secta was attracted to the white paper flowers baited with a blend of benzaldehyde and methyl salicylate. The addition of oligosulfides to flowers of Jaborosa integrifolia in the wild resulted in the nearly immediate attraction of saprophyll flies, mainly Califoridae, to the flowers. Finally, we tested for the importance of oligosulfides and flower coloration to attract flies using white and black artificial flowers. Saprophyllus fly species, mainly musids, califorids, and sarcophagids, were observed approaching and landing on the arrays of black and white artificial flowers when scented baits were present either within or outside of the flower array. Interestingly, the presence of white artificial flowers resulted in significantly more landings than the black flowers, that are perceived as different by the visual system of the flies. So far was a brief introduction about our previous studies with fragrances. Now I will show you a work we did in collaboration with Florencia Soteras, Ana Ibáñez, Stephen Detter, Andrea Coacucci, and Robert Raguso that was recently published in Plants. In this study, we used 19 bioclimatic variables to adjust ecological niche models in geographical space. We obtained a total of 344 precise graphical coordinates of presence for 21 Jaborosa species, 289 occurrence data for Lowland clade, and 55 occurrence data for the Lowland clade. To capture the variation among environmental data, we conducted a principal component analysis using the environmental variables that most contributed to the ecological niche models of both clades, and extracted the first principal component. These PC1 scores capture environmental variation and were used in subsequent analysis. The darkest color shades in the map represent the areas with the highest suitability values for the Andean clade in blue and for the subtropical lowland clade in red. The areas of the highest climatic suitability for the Andean clade were the Andean and mountain regions, while for the subtropical lowland clade was the Pampean region. Temperature and humidity contributed 94% to the Andean clade, while altitude and specific humidity seasonality contributed 90% to the subtropical lowland clade. A total of 84 compounds were identified from trapped floral headspace of the 8 studied species that were grouped into 7 biosynthetical chemical classes that are shown in different colors here. In this network, the black square in the top represent the 8 species studied, while the color squares in the bottom represent the chemical compounds identified in the blends. Bar width correspond to the relative percentage of a specific compound in the blend. Chemical complexity and compound diversity of floral scent vary between clades. 2 chemical classes, aromatics and sesquiterpenes, dominated the volatile blends produced by 3 hawkmoth-pollinated Jaborosa species, and 6 chemical classes, aliphatic, aromatics, irregular terpenoids, monoterpenes, sesquiterpenes, nitrogenous, and sulfurous compounds were represented in the floral scent of the 5 species sampled from the Andean clade. In addition to this clear association between chemical classes and clades, our data also reveal high variation in chemical composition among species, with most of the floral compounds being species-specific. Flowers pollinated by saprophyllus flies emitted fetid odor recalling feces, rotting meat, or carcasses. Although such odors are perceived by humans as strong, they are rarely recorded in large amounts, suggesting that flies are highly sensitive to their presence. In contrast, night-blooming flowers pollinated by hawkmoths emitted powerful perfume, like scents characteristics of jasmines and gardenias, known to elicit hawkmoth attraction. We performed a non-metric multidimensional scaling to analyze differences in scent profiles among Jaborosa species and pollination modes. We projected the tree in the ordination to take into account the phylogenetic relationship among species. White circles show the 3 species from the subtropical lowland clade, and gray circles the 5 species from the Andean clade. Some compounds that are important explaining differences between fluorescent samples are shown. Blue arrows show the influence of environmental variables in the scent space. This phylo-scent space show that the 2 Jaborosa clades differ significantly in floral scent chemical composition. 6 environmental variables help to explain the differences. Maximum temperature of the warmest month and humidity were associated with the samples from the subtropical lowland clade, whereas temperature, seasonality, altitude, and diurnal range temperature were associated with the samples from the Andean clade. As I previously showed for Corolla color, we also reconstructed floral scent and pollination mode evolution in the genus. Phylogenetic relationships were projected in the space defined by floral scent profile from the previous NMDS analysis. In the upper graph is represented the axis 1, and in the lower graph it represented the axis 2 from the previous scent ordination. Pollination by nocturnal moth is represented in green, generalized by small insects in yellow, and saprophyllus flies in black. Chemical classes of the floral box that are important explaining the ordination are shown on the right. Ancestral reconstructions of pollination modes were contingent with changes in the floral scent profiles. These changes varied from relatively simple profiles dominated by aromatic and sesquiterpenes in the hawkmoth-pollinated species to complex mixtures including sulfurous, ring nitrogenous, monoterpene, aliphatic, and irregular terpenoid compounds in the saprophyllus fly-pollinated species. The use of a combined approach based on complementary modes of chemical analysis, multivariate analysis of the resulting scent profiles, ecological niche modeling to estimate environmental suitability areas, ancestral reconstruction of floral scent and pollination modes allowed us to conclude that the ability of Jaborosa plants to colonize newly formed environment during Andean orogeny and the ecological changes that follow were concomitant with significant changes in flower scent composition, several of which are known to trigger specific responses in nocturnal nectar-feeding hawkmoths and diurnal saprophyllus flies. Working with Jaborosa has been a long journey, and many people have helped me a lot during these years. Thank you to all of them and to you for listening.
Thank you, Marcela. That was amazing. Like, yeah, so many results and such a good story. Yeah, about telling about the story of the scents and the evolution of the color. That has been amazing. Like, congratulations. It's really, really nice. And well, first we have a question of Federico Roda.
Okay.
You can remember that you can do it in Spanish or English. It's fine.
Hi, yes, it was a very, very nice talk. I agree. I have a couple of questions. So it's about the flowers that are pollinated by the flies. So yes. First, they seem to have a lot of trichomes. So what yes. is the role of the trichomes, both visually, chemically, physically in the pollination? Do you know?
I don't know. It's something very interesting to look. Some hairs I know that offer like sugar because I squeeze the flower against the glucose stick strips to test for sugar presence, and in some species they have sugar in the trichomes, and flies are like there. but I don't know if there is something to study. But they are present mostly in the Andean clade. The 3 species pollinated by hawkmoths do not have the hairs.
Thanks a lot. And the other is about the black color. Is it caused by anthocyanins? Do you know the pigments? That
I don't know, I suppose, but I'm not sure. I haven't checked for that.
Okay, thank you very much.
Thank you, Fede. And Stacy also has a question.
Okay, I'm sorry, my camera looks terrible, but thanks, Marcela. It's such a beautiful group, and I'm just interested to try and figure out, why some clades in the Solanaceae have some transitions and others don't. So, yes. Aroma that I study, it doesn't have any transitions to specialist moth pollination or fly. But then like Jaborosa, nothing went to hummingbird pollination.
Yes.
So I just wonder if we zoom, this is terrible. I can't remember what genera are around Jaborosa, but like if we zoom out, are there other hummingbird species, or do you think it's like, what do you think's going on there? They just geographically aren't where there are lots of hummingbirds?
Yes, I don't know, Rocio maybe knows, but Jaborosa is near to Latua, but I don't know the support. And yes, I'm not sure why, because Salpicroa, but it's not close to Jaborosa, has many species pollinated by hummingbirds. It's like, I don't know, it was there in the right moment.
It just makes me think that maybe, some types of transitions work and then some you get stuck in. Like once you've become fly specialist, you're just kind of stuck there, or
yes.
Once you become moth specialist. Anyway, really, really awesome. Thank you.
Sorry, I was just checking like the phylogeny. Yes, it's Latua, and that was monotypic, and there is no much information about Latua.
No, Latua is hummingbird pollinated, I think. Latua is from the southern part, yes, from the southern part, and
Valdivia.
Probably hummingbird pollinated.
Yeah, it's probably, but I don't know. I haven't seen any study like actually checking that. But yeah.
No.
That's interesting, yeah. And there is also a question of Kelsey Byers. I don't know if you want to do the question or I do it just for you.
I can say it if you can hear me.
Okay, yeah, we can hear you.
Okay, so these behavioral studies are super cool, right? So you supplemented integraphilia with dimethyl sulfide and dimethyl trisulfide, and that quickly attracts the flies. Does the opposite happen if you supplement Rotaceae with the benzenoids and farnesol and so on? Does that attract hawkmoths, or is it the hawkmoth visitation just too rare to get data?
I haven't checked that. It's a missing step in the study. I'm not sure because moths need like a visual cue to be close to the flowers, and I'm not sure about flowers. It is something to check. I don't know.
Yeah, it would be, do you know in like flight chambers, would the hawkmoths go to a white flower that's scented with dimethyl disulfide?
Yeah.
Okay, cool, thank you.
Thank you, Kelsey. And yes, there is a lot of, well, of course, congratulations, great talk. Yes, Kelsey was saying also that she has seen the paper and she, yes, haven't had a chance to read it, but it's really grateful to see it here. Loretta Freitas is also saying wonderful work. And I don't know if there is any other questions for Marcela.
I'm reading the last part of Kelsey's question about hawkmoth visits. I have to share, let's see if I can. One slide I prepared just in case. I'm not sure if I am
Yeah, now we can see your presentation.
Is there?
Yes, cool.
Okay. So it's extremely rare to see hawkmoth in the field. It's like you, in Jaborosa, I never saw one. It's like you can be like many hours and it's not possible to see it. When I went to Robert Raguso lab, we put the plant and the moth in a cage and they immediately went to the flowers, and it was awesome to see just there, but in the field it's extremely rare. And during my doctorate, I use light trapping, and I collect like almost 658 hawkmoths, like a lot, and took pollen samples from the proboscis, and only 7 individuals of all of them were carrying Jaborosa integrifolia pollen. And all the individuals, the 7 individuals, belong to one species, Manduca tucumana. But to see hawkmoth in the field, in the Jaborosa, is almost impossible. The other 2 species, Jaborosa runcinata, that have like short tubes, I only saw scales, because when the moth is visiting the flower, like the hairy stay there in the corolla, and so I check in runcinata and odoneliana with a lens the presence of scale, but I never watch hawkmoth visiting that 2 species, only scales. And odoneliana is a very rare plant, and the flowers also, because they have like a very short stigma in the bottom of the tube. And I think we are collecting flowers from the same plants for years because it's the Hunziker location. I don't know, it's very strange plant.
Cool, thank you so much. That's a lot of effort. I'm really impressed.
Yeah, it's amazing. That's right. So I do have a question that it's kind of a silly one, because I'm just curiosity. What is the next step? Like, what other questions?
I don't know. I'm working. Jaborosa took a long time, like a decade, to finish. It was my postdoctoral project, and then continue at the beginning of my research at la carrera. And now I'm working actually with Eugenia Drevniak, who is a doctoral student in Bergen AC. So we are always looking for floral traits and determining pollinator interactions, but it's like there are so many things to do with Jaborosa, but they are far away, and when you are there, they are not in bloom, and it's difficult to work with Jaborosa in the field. But I think the most promising part would be working with intraspecific variation like Jaborosa reflexa, because there is a huge variation among populations, and I think that is something very interesting to look at, different floral traits and different pollinator assemblages, because it's widely distributed in the Patagonia. So I think that would be a really nice thing to check.
That's awesome. Yeah, and I know, like, yes, there's much variation about flowers, and their scent, the color is amazing. But what about the fruits? There is something exciting about the fruits of Jaborosa?
The fruits are really smelly. I have a picture. Really smelly. I think that probably they are dispersed by mammals, but I don't know if someone studied that.
No one is never interested in fruits of seeds.
It's difficult. That one is the smell is like, I don't know, it's like a tropical fruit. I had like runcinata fruits in the lab, and they smell like for distance. It's really smell.
Wow, yeah, that's amazing.
This is Jaborosa reflexa, but all of them look similar.
Yeah, and the seeds. Well, yes, I'll write you about the seeds probably later. Yeah. That's great. Yeah, and Stacey's also saying crazy fruit. Yeah. Okay, so I think if there is no more questions, be free if you have any other question to ask. Otherwise, we are finishing the seminar. And next week, next Friday, we are going to have Lynn Boss and Ellen Dean talking about the diversity in the genus Lyciantes in Mexico and Central America. So stay tuned for next seminar and see you very soon. Thank you very much, Marcela.